Então, continuando as atividades, agora nós convidamos o Dr. Aloysio Borém de Oliveira para moderar a palestra do Dr. Elliot Heffner. O Dr. Aloysio Borém de Oliveira possui graduação em agronomia pela Universidade Federal de Viçosa, mestrado em genética e melhoramento pela UFV e PhD em genética e melhoramento pela University of Minnesota, onde também fez o pós-doutoramento em genética molecular, trabalhando com silenciamento gênico em soja. Atualmente é professor associado do Departamento de Fitotecnia da Universidade Federal de Viçosa. Por favor, uma salva de palmas para o professor Borém.
Agradecemos a comissão organizadora por essa oportunidade de estar aqui e parabenizamos pela iniciativa de um evento desta envergadura. E vamos passar imediatamente à próxima palestra. Nós estamos 15 minutos atrasados e vocês que são estudantes sabem muito bem que aluno de graduação não espera. Tem uma aula às 16 horas e muito provavelmente a comissão organizadora deve ter que arranjar uma outra pessoa para moderar ao final. Hoje nós temos o privilégio, o prazer de estar recebendo uma autoridade em seleção genômica. Trata-se do Dr. Elliot Heffner. O Dr. Elliot Heffner possui a graduação em agroecologia pela Pennsylvania State University, o PhD em melhoramento de plantas e genética pela Cornell University e atualmente é melhorista de plantas de milho do Pond Pioneer. Com isso, eu convido o Dr. Heffner a assumir a palavra nesse momento. Would you please come, Dr. Heffner?
Boa tarde. That's going to be the extent of the Portuguese. I apologize. So I just want to thank the organizers. This is a great opportunity to come to Brazil. I very much love the country and love the people. I thank the students. They did a fantastic job putting this together. Leonardo and Andrea have been great hosts. Thanks for all the help with the travel and also the tours around campus. It's been very enjoyable. And I've enjoyed the talks this morning, and I hope to expand on those talks and talk a little bit about what we're doing at DuPont Pioneer in the area of genomic selection, and also in the general area of how we apply markers in our breeding programs. So with that, let's get started. Okay. So, there we go. So to just expand on the introduction a little bit, I just wanted to have a couple notes about what I actually do at DuPont. And so I'm a product development and raised breeder for the company. And so this is a map of the Midwest in the United States. So here we are in Iowa. And this yellow area that you see there is the region that I'm primarily responsible for. And so my breeding station is located in Dallas Center, Iowa, which is a small town near the headquarters where DuPont Pioneer is located. And in terms of what I breed for a relative maturity of 106 to 110 days in maize, which is quite early in comparison to what is grown in Brazil. And when we talk about how we test hybrids for my particular breeding program, I test throughout the middle maturity of maize in the United States. And so if we look at this circle here, that is well over 1,000 kilometers in terms of its width. And so my focus is certainly developing inbreds and hybrids for that region, but in my breeding program I also focus on the program and across the company as a whole. And then talk about the need of precision phenotyping, the goals of marker-enhanced breeding, and how we use marker-assisted selection in the traditional form and use genomic selection in the recent years. And then a key point, I think, is talking about ongoing research. So what are our questions and where are we going forward? Because we've been using genomic selection for a number of years now, but that's not to say that we're finished asking questions. In fact, a whole new host of questions are now appearing. And so there's a lot of need for both public and private research in this area to continue. So, to start, global crop consumption, maybe you can't see the numbers here, but we're talking different crops. Maize, which is my primary responsibility and my focus in my breeding program, is 40%. Global production goals, we know the population is increasing dramatically, 9 billion people by 2050, and so we need to basically double our production in order to get there. And we also know that our markets can be unstable at times. So we need not only to increase our food supply, but to make it more stable. And the slide that Tabaré has already shown today, Pioneer is focused on these goals. We have more than 4,000 researchers working at Pioneer, and we're a global company for sure. So this is a very general schematic, but when we look at how our research goal in developing better products, we start with an elite germplasm pool, which I'll talk about today, and we're combining conventional breeding methods, molecular breeding, and transgenic to reach these goals. We assemble that biological information, thank you, and we're using that information to create superior products. So, for those who aren't so familiar with maize in North America, this is a very typical slide that we show, because hybrid corn, the hybrid maize, starts in this area in the early 1900s. And so when we go from open pollinated varieties and we move into hybrids, we can see a dramatic change in yield. And so each year we're at 1 to 2% increase in terms of, and this is bushels per acre. Sorry, but 1% is uniform. So that's the increase that we see as we go over time. And so how do we increase this rate beyond what we're already at? And what the answer is, we need to increase our genetic. And so as we look at this, most of the progress that we see in that graph is actually from traditional field breeding, which is plant, measure, and select, okay? And quite honestly, we have new tools, but it's still a lot of the progress is from conventional breeding. And so now we have planters where we don't even have anyone on the back dumping seed packets. We have precision phenotyping tools, and this is the glorious wind machine. So wind is a very important factor for us because it knocks plants over, which is called lodging. It also can snap plants in half. And so this is a machine that was developed at Pioneer that produced winds well over 100 miles an hour. So let's say, well over 150 kilometers per hour. And we also have precision combines that can take multiple plots at a time. So new tools, we can go much faster and measure many more plots, but this is still considered, I would say, traditional field breeding. Where we're going now and have been for the last decade is really integrating these areas of genomics, and the environment. And this is where we see the big promise for increasing gain. And it's not just the question of understanding these different portions, it's delivering it in a selectable format. And so as a breeding company, we need to understand these areas, but the key is then to translate that information to our breeding community so that we can use them in our breeding program. And we have an extensive software group and that investigates and looks at how we visualize this information to make it into a selectable unit. So molecular characterization in germplasm. So we call this the brain, the two lobes of the brain at Pioneer. And what I'm showing here is actually a graphical representation of the history of Pioneer germplasm. So if you remember from the graphic that I showed before in selection, when we started in the early 1920s, we're talking about open pollinating. So we're not inbreeding, we're not creating hybrids. But as we form the heterotic pools, we then select these pools to where we operate now in our current germplasm. And so we move from the founders into where we are today. And we can trace back to the founders using just pedigree information. So this doesn't require molecular tools at all. We have an extensive pedigree system, and if I create a new inbred today, I can put that in our system, and I can trace it all the way back to the 1920s and look at the founders that are included in that new inbred. And so this is just a little slice of this pedigree galaxy, as we call it. And this is just a representation of all the possible inbreds or new recombinants that we would produce in the breeding program. And so another way to look at this is we can select an inbred and then start to look at molecular characterization. So here are the 10 chromosomes for maize. And this is just a marker profile in a particular inbred. And you can see that this is physical distance because you see a clustering of markers in this region. And the region here is, actually it's physical, this is genetic. And what you see here is a clustering of markers in the centromeres. And so we can take a characterization of one single inbred, but we can also look at a particular region. So here I just have highlighted the first 20 centimorgans on chromosome 1. And I can pick a whole subsection of the pedigree galaxy. And I can say, how do these inbreds compare to each other? So this is an identity and state matrix that just shows a comparison of different inbreds on the top and different markers here. So that probably doesn't mean a whole lot to us, but what we can see here is the comparison, and then we can take that to the next level. So we can take this identity and state information, which is just SNP marker calls, and we can put this onto the backbone. So here's a pedigree of an inbred, and we trace it all the way back to the founders. And if we pick a segment here, say 10 centimorgans in size, we can look at what we have today, and we can then track that identity and state through the pedigree. But it's quite difficult to visualize. So what we can also use is identity by descent information. So this is a haplotype view of converting that identity and state information into a haplotype. And what we can see here is we have a haplotype in the bottom that shows a recombination of 2 founder haplotypes from the beginning of our germplasm. And what we can do is we can trace this back through the history, and we can actually see where that original recombination occurred, and then we can see the inheritance of this recombinant haplotype. And so similar in terms of the comparison I showed before, where we had this matrix of identity and state information, which was just the SNP calls, we can look at comparisons across inbreds in identity by descent look, which is haplotype, and here's the look here, where we can see I have marked founder lines, so 3 founders that we had in the 1920s, and then we can see what our current germplasm looks like and how that's combined. So we can look at areas of where the genome is fixed, and we can look at where the genome is still segregated in this particular sample of inbred. And then we can look at all of its progeny as well. So this is basically just looking at more individuals and looking at all the new inbreds or double haploids that are coming through our system. We can use this information to also search for novel recombinant events. So this is just a recombination finder tool that we can use. And I can say in a particular area that I'm very interested in, I want to search all the inbreds in the company to look for where I had a recombinant in that region. And so I would just define a region. I would say, okay, how many double haploids could I look at? How many of them have a single recombination? How many of them have 2 recombinations? And so on. And so in this particular case, in this sample, you can see I don't have any of the 3 or more recombinants, but you can see all the single and double recombinants in this region. So precision phenotyping. Molecular tools are not very useful if you can't link them to phenotypes. The characterization is very important, but where we really want to go is to use these as predictive And so we also need to have a precision phenotyping system on these inbreds. And here is just an image of the Midwest in North America. And you can see here, this is actually a yield advantage comparison. So what I would have done in this case is I say, okay, on hybrid X, I want to compare it across all the hybrids in our company growing this year. And then I want to look at, based on geography, if this particular hybrid was positive or negative in terms of yield. And this is just a summary, which really displays the extensive phenotypic network that we have and the ability to genotype or to phenotype a hybrid throughout the country. So we characterize our germplasm. We use detailed experimental design because we try to limit noise as much as possible. Special phenotyping to also increase heritability and reduce noise. Managed environments like for drought. And trait dissection. So we want to not only measure our traits, but we want to understand our traits and dissect them into composite traits if possible. So what I've done so far is talk a lot about descriptive activities. And what I mean by descriptive activities, we run a SNP chip and we look at what markers are there. Or we go into the field and we measure. But that's descriptive. And we say, okay, we measure and then we select. But the real area that we want to be operating in is taking descriptive information and making it predictive. And so we're leveraging a descriptive pipeline to enable prediction. So we're taking our pedigree information, we're taking our genotypic information and our phenotype, and putting it into the system to create genetic signal and use that genetic signal throughout our breeding program. So we've already seen this formula today, and so I'll go quickly because Matt has done a nice job at explaining this, but I will just talk about how it relates to maize breeding or plant breeding program in general. And so we want to increase our selection intensity, and we actually do have opportunities for changing genetic variants in our situation because we can evaluate more individuals and more populations, perhaps work into germplasm that we couldn't work in before because we now are expanding our breeding programs. And thus we can think about it, enhancing genetic variation. Increase our selection accuracy by enhancing estimates of true genetic value. And of course, the biggest changes that we can make in our breeding program is in the denominator of the formula. So reducing time and cost. So how can we reduce the cost of estimating a breeding value and reduce the parent cycle time, which is the time from pairing to progeny and then back to becoming the parent of the next generation. So this is quite familiar to many of you, so I'll go quickly, but I just want to make sure that we're all on the same page. And so when we talk about conventional marker assisted selection, we create a segregating population for the trait of interest. And plants, we have a unique opportunity for this because we can take two parents and make a specific crop for our trait of interest. We genotype and phenotype. We can identify and validate significant genomic regions of QTL, and then employ marker-assisted selection for those few target regions for those selected candidates. So here we identify two particular regions. We classify the haplotype, whether it's positive or negative, and then we can make selections in our breeding program. And so, has this worked? Yes, it has worked. It has worked quite well, especially for traits like disease, where we have major effect loci that are controlled by a few large effect and significant QTL. And so significant, or very successful examples rather, are gray leaf spot and northern cord leaf blight. We've done a very good job in using conventional marker-assisted selection for these tools. But there's an issue, and it was mentioned earlier today, the Beavis effect. So as we start to think about our other traits, they tend to be highly polygenic or controlled by hundreds of genes. And so when we think about these traits that are extremely important, like yield, for example, can we think, or even guess that we would do a good job with conventional marker-assisted selection to predict these traits? And the answer is probably not, because it was a system that's built for a few large effect loci, and these traits are controlled by hundreds of small effect loci. And so this is where we need to think about not just the areas that fall above the significance threshold, but also fall below the significance threshold. We know there's error there, but we also know there's true value. There's true effects that are in that zone. So genomic selection, it's already been covered today. I'll just go along with the conventional markers. This is selection format, just so we can see the differences. And it's not so different. I mean, we also use segregating populations for the trait of interest. We evaluate genotype and phenotype those populations. And here's the difference. So as we've discussed earlier today, or have been discussed earlier today, the difference is we're looking at a genome-wide effect. So we're estimating all the effects across the genome, and then we sum these marker effects to come up with a value. So instead of saying I have a positive haplotype in region 1, and a positive haplotype in region 2, I'm saying what is just the overall sum of all the positive and negative regions, and then I get my estimated region. And so a big question would be, well, what is our prediction accuracy in maize at Pioneer? And so I'm just going to give one example, which is for grain moisture, which maturity. And in this example, what we did was we have a large data set where we subsampled, and we subsample a training set. So these letters that represent inbred. And so we're looking at the progeny between each of these crosses as the training set. And then the prediction set is another cross. Here in this case, just this example, 88 inbred. And a key thing to point out here is that the two parents of this cross are actually not in the training set. And so if G and H were also present in the training set, you'd actually expect accuracy to be even higher. But what we want to say in this example is, what if we're predicting a new cross that we've never seen before? What could we do in that kind of scenario? And so we look at predicted versus observed. And here we see a summary of 1,600 data sets. Okay, so instead of just showing a mean accuracy across 1,600 cross-validation, what I'm showing here is the distribution of all those accuracies. And so there's a lot that you can look at this kind of scenario, because we have some populations that don't do very well. The accuracy is quite low, or it hovers around 0. But overall, we see, in fact, a very positive effect of grain moisture, and we actually do quite a good job of predicting this trait. And so it's very valuable to use genomic selection. So a more informative thing to say versus for this case, instead of going for each individual accuracies for each trait in each scenario, I think a more informative to say is, well, how good is genomic selection at Pioneer, and how valuable is it? And the reality is that it's integral in our breeding program. And we use genomic selection throughout the entire process of breeding at Pioneer, when we go to inbred and cross prediction, all the way through the hybrid development pipeline. And so, with that said, I want to give a few examples of how we actually use genomic selection in our breeding program. And the first is to create breeding crosses. So we not only use pedigree values, pedigree information and breeding values of the parents, but we also use genomic selection values. And so here I just have a matrix of looking at all the possible inbreds that we can make crosses of. So these would be all the possible crosses. And then we use predictions. The way we do that is we actually simulate the progeny of each of these crosses and then make predictions. So we can predict the mean of each cross and also the variance. And we can use this information to select populations before we ever make them. But an important point here is that you can already do a very good job with phenotypic information and breeding values to do this task. Okay? What you can't do with pedigree information is discriminate between an inbred that's a full sit, or saying two inbreds from the same cross or from the same parents. What you can do with markers is make a discrimination between them. Because the expectation is 50-50. Right? Those two individuals are the same based on their parents. But with markers, we can actually make selections within families. And that, for us, is a really big point. And this is a new area that genomic selection takes us. So before we ever get a phenotype, we can make selections in the family. And so when we get these double haploids back, we make selections here, and we can see that we use it for a lot of different traits in the breeding program. So we talked about cross prediction, making the initial cross, then getting the inbreds back and making predictions on those inbreds and making selections between them with genomic selection. And we can also use it to make hybrid predictions. So again, if we look at all the possible combinations of hybrids, here you can see one heterotic pool and the other heterotic pool. And if we combine those inbreds, here would be all the possible hybrids. And then we can make selections before we go to the field. So what we're doing is we're expanding our breeding funnel or our virtual breeding program. So it's impossible for us to phenotype all these hybrids. But what is possible for us is to genotype them all, make predictions on the combinations, and then select the favorable tail, and those go to the field for additional evaluation at the phenotype level. So I mentioned that genomic selection is integral to pioneer maize breeding. But we're now moving into the question of optimization. So we're assembling a lot of data. We know that genomic selection is advantageous for our breeding program. But we want to now look at how can we improve what we're doing. And so optimization is where we're going. So one of the big questions is what populations are we putting into our training model? So we can create many more populations of individuals than we can phenotype. So there's a question of which ones do we actually want to put in the field to retrain our models to use as the base for these predictions. And so which populations or which connections of populations are going to be the best for us to put in the training model? And then based on those crosses that we choose, so say these are the ones that we decided on. How many progeny for each of those crosses, and which progeny do we want to put in the training population? So if you have genotypic information on all these individuals, how do we select which ones we should put into the training set in order to train for the next generation, and which ones do we just leave behind? Whether we say we're selecting against them and moving them out of the breeding program, or potentially keeping them there and putting them in the breeding programs, but not putting them into the training data set, the training population. So I mentioned before that we can do hybrid prediction. Well, this is the testable unit of a breeding program for us. Hybrids are what we care about. And so when I say let's decide which inbreds we want to put in the training population, you have to take that a next step forward, because what we put in the field is not inbreds for yield trials. We actually put hybrids. So now once you've said, okay, this is, let's say, the inbred that I wanted to select to put into the model, what hybrids do I want to make with that? So it's not only selecting the inbreds in one heterotic pool and the other, it's then saying, now that I've selected those inbreds, what are all the combinations that I want to make to put them into the field? And one of the things that Matt and I were actually talking about earlier during this visit is which environment. So we're asking these questions of what genotypes we want to put in the training population, but the next step is the environment. Because in plant breeding, we know we have massive genotype by environment interactions. And so as we look at all the possibilities in the middle of North America, and where I am with my breeding station, where do I want to take these hybrids that I've selected to put in my training population? Where do I want to put them in those environments? And do I replicate the same individuals across all these environments, or do I take subsamples and put these hybrids in this area and a different set of inbreds or hybrids in this area? So there's a lot of optimization questions that we ask in terms of how these training populations are formed, and then how we test them. So the testing strategy is extremely important for maximizing the information that we get in the training set. And we're not just doing this in a single year. We don't train a data set and estimate marker effects and then throw away that information the next year. We're assembling training information across many years. And so at lunch today, we were talking about this concept of when you first start a genomic selection breeding program, the first thing you're trying to do is just get a lot of data. Because you see, as you get more and more data, your accuracies continue to increase. But once you get to very high numbers of data points, we're asking questions of, well, is there information in there that's more valuable than others? So, and usually through thinking about through selection index theory, how do I rate these years? And how do I say, you know, is 2006 a more important year than 2003? What were the conditions in that year? What are the genetic correlations and the trait correlations that I'm seeing in those years? And how do I compare them and combine them in a training set? And it gets even bigger, right? So we can talk about what environment, and we can talk about what year, but we can also talk about what trait combinations, because these traits don't work in isolation. And so when we think about building training populations and thinking about optimization going forward, we really are asking questions about what are the environment by year, by trait combinations, and population combinations for that matter, that we want to continue in our pipeline and to use as a breeder to make selections at all the different steps in our breeding program. So I'm just going to go through my conclusions, and I think we have plenty of time. Okay, so I'll go through the conclusions now, and then we can maybe have some time for some questions. So today I described what we're doing at Pioneer in a private sector setting, but I also raised a lot of questions. And so I think it's very important to note that we really need a lot of emphasis and continued research both in the public and private sector, because there's a lot of questions to be asked, and there's a lot of talent in both sectors, and a lot of different crops in both sectors. So I think it's clear that we need to continue research. Molecular tools, the cost of genotyping is dramatically decreasing. The cost of phenotyping is not. It's only increasing. So how are we doing our phenotyping? How can we make it more efficient, and how can we make it more accurate is a really key point. And I don't think there's any question that genomic selection is here, and it's here to stay. And I think it's going to be an increasingly important tool in our breeders' toolbox. And like I said, I think there's, this is my area, so of course I'm excited about it. But I think this group will hopefully agree that there's a lot of exciting and important questions that remain. So acknowledgements, I'm not going to put a lot of names because we're a large organization and there's a lot of people that come together in order to make genomic selection a successful tool for us, both at the individual breeding program level, but also as a global organization across all of our breeding programs. So our breeding technology group, our maize product development, a research information management team that works on all of our software tools to make this possible, and our global molecular technologies group, which allows us to genotype millions of samples in a year. And so with that, I will thank you, muito obrigado, and questions and discussion, please.
Gostaríamos de agradecer ao Dr. Elliot, thank you for coming and give us an excellent lecture. E agora nós temos um tempinho para fazer perguntas, 5 minutos de perguntas, e é um debate com o professor Aloysio Borém. Lembrando que a pergunta pode ser tanto por um papel escrita ou oralmente pelo microfone.
Ok, nós já temos uma pergunta à direita. Enquanto as perguntas chegam à mesa, eu gostaria de escutar um pouco mais do Dr. Heffner sobre o seguinte assunto. You have presented to us that genomic selection is not only a matter of investigation. It's been used in practical plant breeding, and genomics have evolved very much. Now in this scenario, would you consider that genomics the bottleneck in the application of this information in plant breeding, or phenomics may be the current bottleneck?
So there's absolutely no question that genomics and phenotyping is more of the bottleneck than the genotyping, because as Matt had mentioned, genotyping is becoming increasingly cheap, and I think he said it was 1,000-fold different in 3 years, and the same is true across all species. And so we need people thinking about different marker platforms and different ways of doing this, but really it's in terms of the model performance, we need to continue to look at different models for genomic selection, there's no question. But in practice, with the models that we currently look at, the differences are quite small in most cases. And so the biggest changes that we see in the breeding program is how we put them into practice, how we use genomic selection, and the quality of working with them. And not just the quality of what we've done so far, but really thinking about what's the new way of doing things. What are we missing? What are the things that we can look at to better dissect, better characterize our germplasm, and then put that into the genomic selection pipeline? So clearly, I think phenotyping is probably the biggest.
Temos uma pergunta aqui do Gabriel Luiz. Does Pioneer have some program with U.S. universities? And if you have a program with some American universities, which ones?
So I'm not sure exactly what we mean by program, to be honest, but in terms of relationships, a lot of relationships. So Cabaret is our key person that helps us with those relationships and develops them. And there's many breeders and scientists across our organization that stays very close with the universities. So like the symposium that we have here today, Cabaret mentioned this morning that we're doing this at multiple universities. And even beyond those universities, we visit, we invite them to come visit, and we also give them aid. So we provide fellowships, we provide internships, which were mentioned this morning as well. And the universities are extremely important to us because we want to contribute to the education of the next plant breeders, and we also want to interact with the faculty that are at these universities because there's a lot of ideas that come from them. So it's not just about the presentations that are given today, it's also about visiting the universities, interacting with the universities, and exchanging ideas across faculty, private sector folks, breeders, and the students. And these interactions can't be overstated how important they are, I think, to both sides.
Mais alguma pergunta? Ok, então nesse meio tempo, gostaria de fazer também mais uma pergunta para Dr. Heffner. In the case of hybrid prediction, I'm just wondering if the additive dominant models work well for genomic selection.
So I think as we look at the different models in terms of additivity and dominance, primarily the models that have been used, and I'll speak beyond Pioneer, have been additive in nature. And this is a big area of research, to look at what the additional accuracy that we can get from using the dominance model or hybrid prediction. And then beyond just dominance, looking at interaction terms. So looking at different epistasis terms in terms of incorporating them into predictions. So what I'll say is largely the progress so far that's been in genomic selection is at the additive model nature, and that the importance of dominance terms and in different models that incorporate these terms for prediction is a very active area of research.
The other question that just arrived is about transgenics in the United States. If the U.S. government has been encouraging public acceptance of this GM varieties in the U.S.
I'm not a spokesman for the government, but what I'll say is that the U.S. government is very interested in making sure that products are safe. I think I can say that, and so far that is clearly the case. And I think that we're in an environment where transgenics are a key part of U.S. agriculture, and they have brought a lot of value to the farming community. And transgenic traits are very important to our company, and we sell them widely in the United States and are very important for me.
Mais uma pergunta. Ok, essa seria a última pergunta segundo a comissão organizadora. Ok, a pergunta diz o seguinte: how is the information on pedigrees and molecular markers being applied in planning the breeding experiments?
So the evaluation of pedigrees to create experiments, to create breeding populations, along with marker information, is one of the areas that I spend most of my time as a plant breeder. So the selection of crosses to put into the pipeline, into the breeding pipeline, is absolutely critical. And anyone that's been in plant breeding for a while or is studying it for the first time, I think quickly finds out that quite possibly the most challenging part of plant breeding is picking populations. Because if you can do that correctly, you put yourself in a very good position for creating a lot of genetic gain. And so a lot of the tools that I showed some screenshots of are aimed at doing just that activity. It's integrating pedigree information and marker information to display it in a usable format for breeders to make their population selections and determine what process they want to work on the breeding program. So I'll just say that it's a big area of what we do as plant breeders, and we are using molecular information, like the haplotype pictures that I showed, and looking at the different haplotype contrasts of one parent versus another, predicting the genetic variance and mean that we would expect from that cross. We use all that information to make those decisions.
Em nome do colegiado dos professores do curso de pós-graduação em Genética e Melhoramento, eu tomo esse minuto para fazer um agradecimento especial à Pioneer por ter viabilizado a realização deste evento, nos brindado com excelentes palestras, e na pessoa do meu amigo Tabaré, meu colega de doutoramento, fizemos disciplinas juntos, defendemos tese aproximadamente na mesma época. Eu gostaria de que ele externasse aos superiores dele o nosso apreço como Universidade Federal de Viçosa por esta grande oportunidade de estarmos reunindo este grande número de interessados em melhoramento de plantas. Então, nosso muito obrigado. Thank you very much.
Nós também, do Dia Melhor, agradecemos a palestra do professor, do Dr. Elliot, e também agradecemos o professor Aloysio Borém, que deixou de fazer alguns dos seus afazeres para estar aqui junto conosco. Muito obrigado.